All right, thank you very much, especially to Dr. Fries and the organizers of today's great symposium for giving me the opportunity to really share our work, which I never thought would be shared within the glyco community, to be honest. My mentor, Zev Ronai, is actually a melanoma biologist, and so we study melanoma and transcription. We are not a glyco lab, and we were actually very surprised to sort of stumble in our studies into the world of glycobiology. And so today's talk will actually be about how we took our project, which focused on an oncogenic transcription factor, and eventually ended up in fucosylation. So I'd first like to start by giving some brief background on the key transcription factor that we've been studying, or my mentor has been studying for the better part of over 15 years now, which is namely ATF2, or activating transcription factor 2. We've had a keen interest in this transcription factor for a long time because it's been at the crux of a lot of controversy in the sense that it is reported to be both oncogenic and tumor suppressive within the same type of cancer. And really, for the longest time, no one knew how that could be possible, that you'd have these two very divergent functions in a transcription factor until this early study came out. What you see here are two histospots, so two microbiopsies off a large-scale human melanoma tissue array, or TMA. And what this study showed was that the nuclear accumulation of ATF2 correlated with advanced metastatic lesions and poor prognosis, whereas in lesions with cytoplasmic ATF2, those lesions correlated often with better prognosis and more primary or benign lesions of melanoma, as well as non-melanoma squamous and basal cell carcinomas. So what this early data suggested to us was that it's possible that the divergent function of ATF2 is dependent on its subcellular localization. That is, that nuclear ATF2 is oncogenic and cytoplasmic is tumor suppressive. So that's where I came into the picture, and when I first started my postdoc, we searched basically for a cytoplasmic function of ATF2 and tried to determine when it goes to the cytoplasm, and if it does, how it might be playing a tumor suppressive role. And to cut a long story short, what we found is that during different types of stresses, for example, genotoxic or chemotherapeutic type stresses, a pool of otherwise nuclear ATF2 would actually change its localization to the cytoplasm, where it would disrupt the hexokinase VDAC complex at the outer mitochondrial membrane. And in this way, actually, ATF2 was promoting mitochondrial leakage during stress or chemo, and thus it was functioning as a tumor suppressor. Right? However, we found that this mechanism overall was not observed in advanced metastatic melanomas. And the reason for that we found is the upregulation of PKC isoform epsilon, which we found to phosphorylate ATF2 and to drive it back to the nucleus. And in this way, ATF2 is able to function as an oncogene and promote survival as well as metastatic spread. I should note that if we generate a synthetic form of phospho-ATF2 that mimics the PKC phosphorylated state, and we put it into cells, that action alone is sufficient to render melanoma cells extremely chemo-resistant and refractory to radiation and all sorts of therapy. Okay. We were able to extend our findings into the clinic. So here are two representative histospots in a more than 400 patient tumor tissue microarray. And what we found was that their expression levels of PKC, so the kinase that activates ATF2, correlates actually with differential survival advantage. So that is to say that in patients that exhibited low levels of PKC, so therefore low levels of active ATF2, those patients had a significantly better survival outcome than patients that had high PKC levels. So at the end of the day, the take-home message is that melanoma patients that exhibit high levels of PKC and nuclear ATF2 tend to associate with poor clinical outcome. Now, there have been numerous other studies that have indicated individually that each of these proteins also correlate with metastasis. So our question really that was the impetus for the story that I'm about to tell you is how does PKC phosphorylated ATF2, or phospho-ATF2, that's I'll refer for the rest of the talk, transcriptionally drive melanoma progression and metastasis. So what downstream transcriptional repertoire is controlled by ATF2 in such a way that melanoma progresses and metastasizes? So as you might suspect from the title earlier, one of the targets that we came upon was a key regulator of cellular fucosylation. So in the story that I'm going to tell you, there are four parts, and the four highlighted points which is the first finding that fucosylation appears to be increased in primary melanomas and decreased in metastatic melanomas. And this trend appears to be controlled by ATF2, which we found can reduce overall fucosylation in the cell by transcriptionally repressing fucokinase, which is the most upstream regulator of the fucose salvage pathway. I'll be referring to it by its better known and more amusingly spelled gene symbol, which is not pronounced by its phonetic pronunciation. Although I have to admit that during our lab meetings, we've preferred the phonetic pronunciation. And what we've also attributed to changes in the transcription of fuc is actually that altered fucosylation states resulting from this mechanism ultimately will affect melanoma adhesion and invasiveness, as well as cell cycle behavior and responsiveness to therapeutics. And lastly, and what I'm most excited to show you is this very recent data that if we give mice melanoma, and then we further supplement them with either dietary fucose or we restore fucosylation by overexpressing this kinase, we can actually blunt melanoma progression and significantly repress metastatic spread. So, fucosylation was new to us completely. I'm sure many of you might already be very familiar with it. But just to give a really brief background, it's a post-translational modification whereby fucose is tagged onto proteins, and it's required for several well-known cellular processes, including blood antigen synthesis, leukocyte homing adhesion. Very interestingly for us that we did not know, because we often think of signal transduction as a matter of phosphorylation, dephosphorylation, and degradation or not, are these key regulators of the signal transduction pathways that we've all come to know and love. And finally, fucosylation is required for pathogen recognition and immune responses. Pathologies that are associated with altered fucosylation are many. There's obviously a huge number of developmental defects associated with altered fucosylation, as is this leukocyte adhesion deficiency, and a variety of mucous membrane-associated pathologies are attributed to altered or defective fucosylation. But in cancer, what's interesting is that the picture is very murky. It's very unclear whether increased or decreased fucosylation is actually good or bad for cancer. What is clear is that there are certain secreted fucosylated serum markers, such as fucosylated alpha-fetoprotein or haptoglobin, which are increased in patients with various types of tumors. And fucosylation has also been implicated in homing and metastasis, particularly of lung cancer. But this has yet to be reported for example, melanoma. Really quickly, how fucosylation works. Fucosylation either enters the cell from dietary sources or is recycled from fucosylated pre-existing proteins within the cell, but it is not usable until it's first phosphorylated by the fucokinase and then GDP coupled by FPGT, rendering it into a GDP-coupled fucose. Now, this short but very crucial pathway is otherwise known as the fucose salvage pathway. The GDP fucose is then shuttled into the ER-Golgi apparatus, where it's tagged onto proteins by a variety of different fucosyl transferases, and upon appropriate folding and whatnot processing, it's then shunted up to the surface or wherever it's going to go. I'm also going to briefly mention that there's this de novo synthesis pathway whereby GDP mannose is converted into GDP fucose, but for simplicity's sake and a few other reasons, I'm just going to focus on this pathway for this talk, and in particular because it's the one that's regulated by our transcription factor. So the remainder of this talk is going to be split into 4 very short sections. So I'm first going to get into how exactly it is that we identified fucokinase, or fuc, as one of the downstream targets of this oncogene. So essentially what we did was we took a melanoma cell line with low ATF2 and low PKC and depleted ATF2 and replaced it with either a synthetically point-mutated transcriptionally inert ATF2 or a constitutively active form of ATF2 that mimics a PKC phosphorylated form. We essentially looked at the downstream gene expression profiling that was specific just to the activated transcription factor, and then performed gene ontology analysis and found that among the top couple signaling pathways that were identified, that actually fucose biosynthesis came in as number 2, most significantly altered. And if we look in vitro to validate, we found that indeed if we overexpress this active form of ATF2, we can effectively reduce endogenous levels of fuc by about 40 to 50%. So is this effect actually a direct transcriptional effect or not? So when we study transcription, the first thing we do is we chip the gene of interest, and we look to see if ATF2 is binding to its 5' promoter. When we do chip analysis, we find that when we pull down ATF2, indeed it does come down with the Fuchs 5' promoter. And within the Fuchs 5' promoter, upon closer inspection, there's actually 3 predicted ATF2 binding sites. And so, of course, the next step would be to go in and one by one to mutate these elements out to see which one is actually required for ATF2 repression. So if you look at this leftmost line here, that's activity from the full-length wild-type promoter. If we put in phospho-ATF2, we actually can reduce the activity from that promoter by about 50%. If, however, we go through and we mutate one by one the different elements, we find that if we mutate this element number 1, we completely lose the repression that's elicited by ATF2. So these data indicate that, in fact, ATF2 binds to the Fuchs 5' promoter. It binds to it via this most 5' element, and then when it does, it represses expression of this gene. So that's great. We have an in vitro transcriptional mechanism. Does it actually correlate with real fucosylation in cells and in vivo? So the first thing that we did was, and this was another learning experience for our lab, was to figure out how do we measure fucosylation in cells. So we picked the fluorescently conjugated UEA1 lectin, which binds to tertiary structure coupled fucose motifs. And we started to probe a panel of cell lines ranging from primary normal human melanocytes to primary melanomas to metastatic melanomas. And they're arranged here from left to right such that they're either low in ATF2 all the way up to high ATF2 and high Fuchs to low Fuchs, right? And what we found that was interesting is that overall, the primary melanomas seem to exhibit this slight increase in cell surface fucosylation, whereas the metastatic melanomas across the board seem to be reduced. And then if we looked at patient samples in these 2 smaller scale TMAs, so each spot again is a microbiopsy of either patient primary, metastatic, benign, or normal skin sections, stain with UEA1 and then look at the extracted signal, what we find is, again, primary tumors seem to have a slight but significant increase in cell surface fucosylation, whereas metastatics are decreased. So this was interesting to us. The cell lines and the tissues that are anticipated to have high levels of ATF2 have low levels of fucosylation and vice versa. Can we really prove this? So we took a melanoma cell line and overexpressed either control empty vector or phospho-ATF2 or the kinase, a constitutively active kinase, PKC, that activates ATF2. Probed again with the lectin and found about a 40% decrease in fucosylation. Interestingly, if we take the same cell line and we knock down Fuchs, even though the knockdown is about 90%, we still get about only 40% reduction in total fucosylation. So that suggests that the fucosylation that's contributed to the whole cell by this Fuchs-governed pathway is approximately 40%, and likely this residual 60% is contributed by the other pathway. Nonetheless, the question is, does this 40% that's regulated by PKC-ATF2 Fuchs, does it actually hold significant biological function? And so next, that's what we took a look at. So if we take a metastatic melanoma cell line that normally doesn't have very much Fuchs, and we overexpress Fuchs, so this is one of the assays that we do often called the scratch assay. Essentially, we grow melanoma cells to be like a complete skin across the top of a dish, and we make a scratch, image that scratch multiple times over a time course, and look at how fast the cells move back into the middle as a measure of motility. And so what we observed here is that actually if we have the control cells over a 6-hour time course, they move pretty quickly and fill the gap by about 40%. But in contrast, cells that overexpress the Fuchs kinase, that rate of motility is easily halved. So what happens then if we feed the cells fucose instead of overexpress, which is, fairly artificial? Well, if we have a melanoma cell line here that's got high levels of Fuchs, and we feed them fucose, that rate of movement is again halved. And here's that same melanoma cell line here with a little bit lower levels of Fuchs, but the rate is still nonetheless reduced. So if this is the case, that the ability of melanoma cells to express Fuchs can impair their movement, then loss of Fuchs should accelerate movement. Right? So that's exactly what we did here, and that's what we see, which is that control cells close the gap to about 30%. If we lose Fuke either by overexpressing active ATF2, they increase their rate of movement. If we knock down Fuke with 2 different shRNAs, again, they also increase their rate of movement. Now, given what's known about the role of fucosylation in leukocyte adhesion, we posited that perhaps this had to do with something to do with cellular adhesion. So we first perform a very quick and dirty cellular adhesion assay where we just seeded cells and at subsequent time points just washed the cells off to see how many cells were still stuck to the plate. And what we initially saw was that, in fact, the Fuke overexpressing cells significantly stuck to the plate better than the control cells, again suggesting that this is an adhesion-related phenotype. And I think the most striking illustration of that is here to the right, where we've grown melanoma cells as hanging spheroid droplets. So we will actually grow the melanoma cells in upside-down droplets of media, and they'll form these little microtumors. But what happens is that in melanoma cells that are metastatic, they tend not to form nice little spheroid tumors at all. In fact, they tend to try to spread all over the place. And so this is one example, and this is another example. But what you can clearly see is that when we overexpress Fuke, in either case, this weird spread blob structure just sucks right into one very compact, very dense spheroid. Again, supporting the notion that altered fucosylation is affecting cellular adherence in these cells. Okay. Now, if that's the case, and this has some sort of physiological impact in terms of, for example, invasion, I should be able to take a melanoma spheroid that either control or overexpresses Fuke and put it into a more physiological condition, and that should affect invasion. So what we've done here is we took either control spheroids or Fuke overexpressing spheroids and transferred them into matrigel plugs, which mimics a little bit more three-dimensional matrix condition. And immediately what we saw was that the control melanoma cells spread like crazy throughout the matrigel plug, whereas the Fuke overexpressing cells were much more restricted in terms of their invasion into the matrix. So in summary for this part, it seems that by transcriptionally repressing the Fuke kinase, that active ATF2 can effectively reduce cellular adhesion and increase melanoma motility and invasiveness. What was also interesting to us was that the effects didn't just stop at adhesion and invasion. We incidentally tried performing a colony formation assay, which typically is reflective of tumor proliferation. And to our surprise, we actually saw a significant effect when we have melanoma cells that were either transfected with a control empty vector or with the Fuke kinase. So there was a striking difference in the number of colonies and the size of the colonies that were present. So coming from a cell cycle background, originally I jumped on the fax machine and performed a cell cycle fax. And if you just look at these top 2 rows here, these are melanoma cells with high levels of Fuke and low levels of ATF2. If I knock down Fuke in these cells, compared to the cell cycle profile of the controls empty vector transfected cells, there's a slight increase in SG2. Not very impressive until we add chemotherapy. So this is a DNA damaging chemotherapeutic agent, which would normally block cells in G1S. The cells that are knocked down for Fuke have no G1 population. Everything has rushed into SG2, late SG2. Now, if I do the converse and I take melanoma cell lines that have high levels of ATF2 and low to no levels of Fuke, the control cell cycle looks like this. There's G1S, G2, normal distribution. If I overexpress Fuke and restore fucosylation in these cells, you can immediately see that the G2 population goes away. And then when I add chemotherapy, whereas these cells are blocked in G2, the cells that now express Fuke seem to have more G1. So the presence or absence of Fuke and likely fucosylation within the cell is having impact not only on cellular adhesion and invasiveness, but also on cell cycle responsiveness, particularly to chemotherapeutics. Now, this is great. It's all in vitro. Does it actually mean anything in vivo in an animal with a tumor? So I told you that I was going to tell you about dietary fucose and Fuke expression within melanoma tumors and the effects that it has in vivo. So what you're seeing here, so what we did was we took mice and we injected the right back flanks with about 1.25 million melanoma cells. And once those tumors reached a palpable size between 50 to 100 cubic millimeters, we split the mice into 2 cohorts and treated them or fed them either with normal water or with Fucose supplemented water. And so the black line here is the control mice, and what you can see is that within a roughly 4-week period, 3-week period after they became palpable, the tumors were actually 40-fold bigger than they were when they first started. So I have to mention this is a horribly aggressive mouse melanoma cell line. In contrast, those mice that were fed with Fucose that was replaced every 2 weeks into the water, those tumors only developed to be about 20 to 23-fold bigger than when they started. So they're effectively about half the size of the control cohort. And when we looked into the lungs of those mice, we were a little bit surprised by the primary tumor results because we thought that this effect would be predominantly affecting metastasis and invasion. But what we saw here was fairly gratifying, that in the control mice, they had roughly 60 micrometastases per lung per animal, and in those mice that were fed Fucose water, they had about 25. So restoring Fucose to the cells seems to be able to suppress not only metastasis, but also primary tumor spread. Now, one could argue that, okay, I'm dosing the whole system with Fucose. I'm probably stimulating the heck out of the immune system, doing all sorts of things that we don't know. So can we control for that? And that's the next animal model that we did, which was to take the same mouse melanoma cell lines used here and to stably transduce them either with an empty vector or with a Fuc overexpression vector. And again, we saw a very similar decrease, although it wasn't quite as big of a decrease, whereas the control tumors grew to about 25 times the size as when they started. The Fuc overexpressing tumors were only about 17. So it's a slight but significant decrease in primary tumor size. But again, if we look into the lungs of those animals, we get at least 50% reduction in the number of Mets in the lungs of these mice. So hopefully, what I've managed to show you and convince you of today is that Fuc, which mediates Fucosylation as we know, at least in melanoma, mediates cellular adhesion, which then reduces its motility as well as migratory capacity, and in vivo can reduce the extent of tumor development and metastasis. But in more metastatic and advanced melanomas, the upregulation of PKC, which activates ATF2, binds to the Fuc 5' promoter to suppress its expression. And rather than seeing these effects, we instead see an increased motility and invasiveness, somehow an altering of cell cycle dynamics and therapeutic response. And the last few points I'd like to leave you with is that, so again, as I mentioned before, there seems to be this trend that fucosylation is increased in primary melanomas, whereas it's decreased in metastatic melanomas. And now we think we can explain that through this mechanism that's in place. Clearly, this may have some clinical implication in terms of whether or not, for example, we might be able to use Fucose as some sort of adjuvant therapy. And can we possibly screen for diagnostic or prognostic markers that are altered in fucosylation in melanoma that has yet to be explored? I think the most interesting question for me, and it's going to be a major effort in my future lab, is to determine exactly what it is that's being differentially fucosylated between primary and metastatic melanomas. We still do not know what is directly being fucosylated, and that's going to be really important to determine how it is that we're seeing the phenotypes that we're seeing. And should anyone ask me after I finish this talk, I'd be happy to explain to you how we're prepared to do that. So lastly, I'd like to thank, of course, my mentor. He's been a great mentor for the past 7 years of my postdoc. The present and past members of the Ronai Lab, of course, Dr. Fries and his staff for constant help and support and encouragement. Also the Drs. Fukuda and Fukuda for their advice, the SPMRI cores, especially the bioinformatics proteomics facilities, as well as the flow facility, the American Cancer Society and NCI for financial support, as well as my K99 mentor committee, who have been very, very helpful in giving me critical advice through the years. So thank you very much. I'm happy to take any questions.
Question? I'll start first. So your Fucose treated mice, do they have any other?
What was that?
The Fucose treated mice, do they have any other side effects or anything?
Not that we can tell. Okay, so the question is, did the Fucose treatment in the mice elicit any other side effects? And to be honest, this whole mouse model from start to finish was about 5 weeks. This melanoma spreads super, super fast. And we didn't see any differences in weight gain or loss. Not really. I thought I saw them running around a little bit faster the first week they were on Fucose, but then they all seemed to normalize. So it's, Yes.
So, do you think this is unique to melanoma, or what about other?
That's actually an interesting question. So it's a little bit hard to say, but we performed an initial bioinformatics analysis, and what you see here, this is the basically survival outcomes of the TCGA PanCan data set, which is about it's over 4,000 some odd patients, just stratified in terms of survival outcome associated with high or low Fuc expression. And so the red line here is represents patients that have high levels of Fuc. So they clearly seem to have a trend towards better survival outcome. In terms of specific types of cancer that might have been diluted out in here, what I can say is that melanoma seems to have a clear downregulation of this kinase. If, on the other hand, you look at something like breast cancer, there's a very marked upregulation. We don't know why.
In primaries, or what's that?
I haven't stratified the breast cancers yet. Yeah, that was another big TCGA cohort, but that's a good question. Yeah. Yep.
I saw the ATF2 is a transcription factor with the activation domain.
Yes.
How can you repress the binding transcription?
So it has a DNA binding domain, and so ATF2 is one of about 30 some odd AP1 transcription factors, which either hetero or homodimerizes with the other 30 some odd members. So depending on its binding partner, the combination will actually dictate what gene promoters it binds to. Now, just because it's called activating transcription factor, we've actually found that to be rather of a misnomer, because when we look at gene expression profiling, most of the genes that we've found and validated to be true are actually repressed. So it has a DNA binding, a BZIP DNA binding domain. That doesn't necessarily mean that it's only activating. Yeah. Yes. But that doesn't necessarily mean that it's always activating, though.
Also, the CLAP also recognizes the same promoter, right?
So there are consensus sequences, but so there's a TCAG-TCTA sequence, which is the generic AP1 sequence. But depending on the combination, you will get very different varying specificities of what ATF2 plus whatever its binding partner binds to. Yes?
Very cool, sorry. Just can you speculate on anything? I mean, I know you look at it, but
right.
do you think it's like more something as in protein that we oscillate and therefore their own character change so that they're more adhesive, or do you think it's more like a signaling?
All right, so the question is, what about the downstream targets? And actually, thank you for asking that question. We're about to do a labeled fucosylated protein mass spec, whereby we're pulling down all of the fucosylated proteins in the cell and seeing what's being fucosylated. However, that hasn't been done yet. And in the meantime, what I've been doing is working with a bioinformatician. And so what I asked Ali, who used to work here, to do is, first of all, to see if ATF2 and if Fuc are inversely related or inversely correlated in melanoma samples. And she found that in the majority of them, they are, depending on your cutoff for expression. So that's the first thing. Then I asked her, I said, okay, Ali, if you take all of those melanoma patient expression profiles that have high levels of Fuc, right, cluster them all together and give me a consensus profile. Tell me, when you have high levels of Fuc, what other genes are high and what are the genes that are low? So we've started actually characterizing a little bit of this. So what I'm going to show you here. So what Ali found for us was the most anti-correlated gene with Fuc. So when you have really high Fuc, the most downregulated gene is actually ACTR3, which is better known as ARP3 from the ARP2/3 complex, which mediates actin polymerization and nucleation. Which was, I mean, it was almost too good to be true because, okay, we actually might have a pathway that's impacted by fucosylation that would explain what we see in terms of adhesion and migration. So what you see here is a primary melanoma cell line here with high levels of Fuc, low levels of ATF2, either treated with water or with L-fucose. So the red is actually phalloidin stain. And so this is a really weird cell line because if you zoom in, you can actually see that on the periphery, there's a lot of these invatopodia-like structures. But if you treat them just overnight to about 36 hours with a low dose of Fucose, all of those structures go away and the cell sort of spreads out and you lose a lot of the stress fibers. We've also done the same thing. I'm sorry, they should say acetylated GDP-L-fucose. So this synthetic form of L-fucose bypasses the need to have fuc, right? Another primary melanoma cell line with high levels of fuc, again, you have these very marked actin stress fibers. If you treat them with Fucose, the stress fibers largely go away. You have residual cortical actin. If, on the other hand, and probably this is a better example, this is a highly metastatic melanoma cell line with low levels of fuc, high levels of ATF2. So again, you have stress fibers, you've got more stress fibers, because this guy does not have fuc, right? So even if I dose it with Fucose, nothing's going to happen. If, on the other hand, I put this synthetic GDP-L-fucose into the cell, then you get loss of all the stress fibers again. So this is an initial direction that we're going in. Of course, it still doesn't tell us what is actually being fucosylated or not. And so we're going to rely on the mass spec to corroborate what we find here. Yes. We did. If you look at the top row here, this is the left is DAPI stain, so for DNA, and the red is CD45. We still don't know what is infiltrating yet. We just know that it's some sort of immune cell. So on the left is just water treated, the mice that were fed normal water, and on the right is the Fucose supplemented water. And so you can see there's a ton more CD45 positive cells within those tumors. That might be attributable for some of the suppression of primary tumor growth. I think it'll be really important to figure out exactly what types of immune cells are going in. And I suspect they might be NK cells because apparently they have a preference for fucosylated proteins, but we have yet to see. And the same thing in terms of the empty vector or the fuc transduced cells. Again, I don't know if you can see back there, but there's more red dots in the right side than the left side. So there's definitely an what looks like an infiltration, although we have to prove that. So we're on it slowly. Okay.